Tommy is tired and can't think properly because he is hungry. He just heard his stomach growl and wants to eat some cookies. How can this make Tommy happy? He reaches over and eats a cookie. Once the cookie enters Tommy's mouth, it travels through the esophagus into his stomach, where it gets mixed with digestive enzymes that breaks down Tommy's cookies. The broken cookies then enter the gut, where it is mixed with bile and is broken down further. The gut lumen is predominantly lined by enterocytes. There are these tiny finger-like projections called villi on these cells, which help in absorbing nutrients and transporting it to the blood through the blood vessel wall, made up of endothelial cells. Blood from the gut now contains carbohydrates, fats, and other nutrients dissolved in it, which finds its way to the liver. The liver then metabolizes, processes, and stores these nutrients. Hepatocytes of the liver produce bile, which helps in breaking down fats and aids in digestion. Some nutrients return to the blood from the hepatocytes to make their way around the body via different blood vessels, including arteries. The arteries are composed of endothelial cells surrounded by smooth muscle cells to regulate the rate of blood flow towards the brain. Once in the brain, the essential nutrients, mainly glucose, must cross the highly specialized blood vessels to serve the brain tissue, and this transportation is restricted by the blood-brain barrier, which acts as checkpoints that ensure homeostasis and healthy functioning of the brain. The star-shaped cells, called astrocytes, contribute to the blood-brain barrier integrity. The glucose molecule is transported across the blood-brain barrier and finally makes its way to Tommy's neurons, and the neurons can fire. Now Tommy is happy, and he knows the answer to his test. How can we develop organ-specific models to study biological processes? At MIMETAS, we use the organoplate, which is about the size of a cell phone and is a high-throughput platform combining the most recent advances in 3D cell culture and microfluidics. The platform can be exploited to model biological processes, including diseases. Some of these models take the form of perfusible tubes, beneficial for studying the gut or arteries, and vascularized tissue can be grown to study liver function. Additionally, the versatility of the platform allows for co-cultures of different cell types together to make complex models, including the blood-brain barrier.